Hazenberg shares with us a new, from a different angle, an important aspect in allogeneic transplantation, having to do with killer antibodies that are induced against AML, acute myeloid leukemia. So besides the role of T cells for many years in allotransplant, he is now bringing in the B lymphocytes. Dr. Hazenberg, please.
Thank you very much, Professor Hagenbeek, and thank you all for coming here, and thank you for the invitation. Yeah, I'd like to share with you some data on killer antibodies in AML, which is a pretty new and exciting field, I would like to say, I think so. So the purpose of our research, well, as you just heard, acute myeloid leukemia, which is a malignancy, a cancer of the bone marrow, often has a very poor prognosis, of course, in elderly patients, but also younger patients can actually still die from this. So we really need novel approaches to treat this, this disease. And of course, there are many developments, as we just heard, but we decided to look at it from a different angle. So nowadays, most patients with an AML need a bone marrow transplantation, an allogeneic stem cell transplantation, to be cured from their AML. And what happens is, you can see that here, patients get a healthy bone marrow, healthy stem cells from a healthy donor. Those stem cells go home to the bone marrow of the patient and grow out into a healthy, normal hematopoietic system. And in case the leukemia relapses, and what you see now in the body of the little figure are leukemic cells. In case the leukemia relapses, the idea is that the immune system of the donor mounts a potent anti-leukemia response. So it's a form of immunotherapy, but by nature. And so we decided to study this more in detail because we thought we could learn from nature. So it has been shown that allogeneic stem cell transplantations can mount these potent immune responses, and we wondered whether antibodies could be involved in this. So what we did was we selected 3 patients with high-risk leukemia that nevertheless survived after receiving an allogeneic stem cell transplant. We isolated B cells from these patients, and B cells are the cells in our bodies that produce antibodies. And antibodies, what they normally do is they bind to bacteria or viruses or tumor cells and help the immune system killing them. So we immortalized these B cells and we cultured them, and then we took the supernatant of these cultures that contains the antibodies and screened that supernatant for binding to AML. And by doing this and by cloning the B cells that did bind, we were able to retrieve 17 monoclonal antibodies from these 3 patients that very specifically bind AML. So on your left, you can see THP1, which is an AML cell line, and AML M0, that's an example of one of the patients in our clinic. And you can see that the curve shifted to the right, indicating that the antibodies bind to those malignant cells. And the other 2 curves show that there's no binding to healthy cells. Of these 17 antibodies, 7 recognized the same target. And that target was very unexpected, was very surprising, because that target is the U5 SNRP200 protein. This is a large protein, 250 kD, and it's part of the spliceosome complex. And the spliceosome is normally located inside the cell, where it helps, where it cleaves pre-RNA. So every cell has it, but normally it's inside the cell. And what we found was that in AML cells, this protein is actually expressed on the surface of the AML cell and then recognized by these antibodies. So that was novel, and we screened a few more patients, and we found actually that 4 out of 5 patients had mounted such antibodies. So apparently it's a target that is often recognized by the immune system of the donor. Now the second striking observation that we did was that these antibodies, when they interact with that SNRP protein on the cell membrane, actually kill the leukemic cells. So I have this clip, and I hope it works. So what you can see is, so these are 2 leukemic blasts. I'm not sure if you can see it, but they're a little pinkish, and that is the antibody that is colored in pink that binds to these blasts. What you can see is that they get very nervous because of this, and you see that first this one is dying, it actually spits out its nucleus, and then the second one does the same. You can see it happening here. So these 2 cells are now dead. So this is nucleus and this is cell debris surrounding it. So these antibodies kill leukemic blasts when they bind to it, and they also do that in vivo. So we tested this in a mouse model. And they do that via a very specific way. It's called oncosis. It's a non-apoptotic pathway. And that's why we call these antibodies cytotoxic antibodies, because they kill blasts really on their own without any help. So what we learned from nature, from studying patients that through an allogeneic stem cell transplantation cleared their leukemia, is that these patients make leukemia-specific antibodies to really unexpected targets. Now with immunotherapy these days, investigators decide which target should be attacked by the immune system, and we try to help the immune system by making antibody-drug conjugates, for example. But here, we let nature decide what the target would be. And the target was, amongst others, this SNRP200 complex. And interestingly, when these antibodies bind to this complex, they actually kill the target cell. They kill the leukemic cells. And we think that this is a novel phenomenon. We didn't know this before. And we think we can develop this further into novel therapies. I told you 7 out of 17 antibodies recognize the SNRP complex. This afternoon I will present data on one of the other antibodies that we found that is also extremely interesting. And well, if you have nothing to do, perhaps this is a schedule suggestion. So thank you very much for your attention.
Thank you very much for discussing these brand new data. And I particularly like the movie where we see AML cells disintegrate. That's what we want to see more and more, right?
Yeah.
Any questions, please? Yes.
Just one, please. Are you confident that these are very selective, specific antibodies? Do you have enough experience to tell you that?
Yeah. So, first of all, these antibodies were really. So we've also found a lot of antibodies that did bind also AML cells, but also whatever, skin cells or healthy hematopoietic cells, so a-specific binding. So these 17 antibodies were really selected because they only bound acute myeloid leukemic cells, cell lines and blasts, but not healthy cells. Any cell we could get our hands on, so healthy bone marrow, PBMC, skin cells, all sorts of tissues were tested. Yeah.
Perhaps I missed it, but are you trying this yet in vivo, or will you be?
No, so far we have tested it in one mouse model. Interestingly, that was not enough for Nature Medicine. They want us to test it in another mouse model, so we are now doing that in a second mouse model. I think personally that knowing that only one in, well, perhaps I mean, there's so many things that work in mouse models, but then don't work in humans, so I don't see the point. But anyhow, so we have to wait for that. Yeah, so we're doing those experiments right now.
Okay, thank you.
Yeah.
So my question would be, given these killer antibodies and killer antibody-producing cells that you now can isolate, what would be the ideal, the optimal clinical trial that you would do to profit from this finding? Because that's what it's all about, right?
Yeah, right. So there are many patients, I mean, yeah, there are many patients with AML that are just not, they cannot get the normal treatment, like also my colleague explained to you. So elderly patients or patients that already received a lot of chemotherapy, because, and this is like their third relapse or whatever. So I think you would have to start in those type of patients, of course, that have no other options anymore. And then, yeah, in the mice, we saw that these antibodies by themselves kill the leukemia. Of course, you can also think of combination therapy, that you add this antibody to existing forms of chemotherapy. The fact that these antibodies kill by themselves means that you don't have to couple a drug to it, because they already do it. So.
Right. Thank you very much. We are looking forward to your further work.